Hi, everyone. Thank you for joining us today. My name is Nicole Suri, and on behalf of Megan, I'd like to welcome you to this Megan Masterclass with Dr. Alex Wieman. Please use the Q&A button to send in your questions at any time during the Masterclass. There will be time for our guests to answer questions at the end. This Masterclass will be available on demand on the Megan website at a later date. So now it gives me great pleasure to officially hand over to Dr. Alex Wieman to kick off the webinar. Thank you.
Great. Well, thanks so much for the introduction, Nicole, and thanks also for the invitation to speak here. I'm really excited and humbled to be part of the Masterclass series and be able to give one of these talks. I'm Alex Wieman. I'm a Banting postdoctoral fellow here at the Montreal Neurological Institute of McGill University. And the research that I'm going to talk about today is looking at fusing MEG data to look at these, whole brain patterns of macro scale neurophysiology, and fusing these MEG data with other sort of complementary multimodal neuroimaging measures to try to understand the molecular bases of some of the changes that we oftentimes see in the brains of patients with neurodegenerative disorders, so things like Alzheimer's and Parkinson's disease. I'll start with a little bit of background. I won't dive too deep into this because I know a lot of the folks listening to these series are going to understand already kind of what neural oscillations are. There's also been a really fantastic masterclass already given by Fernando Mestu on some of the changes in brain activity that we see in patients with Alzheimer's disease. So I really recommend checking that one out as well if you haven't already. I will spend a little bit more time talking about why I think MEG is a really good tool to study these neurodegenerative disorders before diving into a couple of studies that I think really exemplify what we can do with MEG when we're looking at patients with Alzheimer's and with Parkinson's disease. The first of these studies is going to be focusing on Alzheimer's, so it's going to be focusing on looking at how the accumulation of harmful proteins in the brains of these folks are going to be leading to changes in their brain function, in their neurophysiology. And then the second study will focus instead on Parkinson's disease, and this one's going to go into sort of the neurochemical systems of the brain that we think might be shaping some of these changes in brain activity. So things like neurotransmitter receptors and transporters, and how these neurochemical changes might be leading to changes in brain activity in patients with this disease. After that, I'll go into some conclusions and some next steps that I see this research taking. But I'll start with the background. So when we talk about neurodegenerative disorders, the two most common neurodegenerative disorders are Alzheimer's and Parkinson's disease. These are both disorders of aging. So we say that because they are the highest risk factor for these disorders is old age. These are the most common, Alzheimer's disease is the most common cause of dementia worldwide. And is marked most notably by degeneration of the medial temporal lobes. So we see death of cells in the hippocampus. We know that Alzheimer's disease is related to the accumulation of harmful proteins, things like amyloid beta and phosphorylated tau that accumulate in the cortex and lead to the death of these cells. And Alzheimer's disease is most notably, in terms of clinical features, noted by declines in cognition. So things like memory declines, as well as declines in things like attention and executive functions. Parkinson's disease is the second most common neurodegenerative disorder. It is, instead of being a primarily cognitive disease, it is a movement disorder. So instead of initially seeing problems with memory or with attention function, you typically see these folks presenting with problems moving and maintaining their balance, although there are cognitive impairments that come secondary to those movement problems. It's most notably marked by death of these neuromelanin-containing cells in deep brain structures, such as the substantia nigra. So neuromelanin is just a pigment that's known to be in these cells in these deep brain structures. And the etiology of Parkinson's disease is much less understood, even than Alzheimer's, which we still don't understand that well either. But we know that there's possibly some involvement of these proteins called alpha-synuclein that are misfolding and accumulating at different places in the brain. We also know that the death of those cells in the deep brain are contributing to some of these movement and cognitive problems, but there's really much less known about where the disease is actually starting and how it's progressing in Parkinson's disease. And importantly, both of these disorders have really profound effects on neurophysiology. So again, there's a lot of research in this area. I won't be covering it comprehensively, but I really recommend looking into the work of Fernando Mestu, of Sri Nagarajan, and Kamalini Ranasinghe, as well as others that have really laid the groundwork for a lot of current research in this area. But some of the key features that we see in terms of brain function that are changing in Alzheimer's disease and Parkinson's disease are shown on this slide. So in Alzheimer's disease, we know that slow frequency delta activity as well as faster patterns of alpha frequency signaling are related to the accumulation of sort of the pathological hallmarks of the disease, being the accumulation again of these amyloid beta plaques and these phosphorylated tau tangles across the brain. A really good example of that comes from a recent study by Kamalini Ranasinghe et al. In San Francisco that was published in 2020. So I have a figure from that kind of representing it over here on the right. What they did is they recorded using MEG the amount of alpha activity in the brains of patients who have clinically defined Alzheimer's disease. So that's what you see on the x-axis down here, with more alpha synchrony being farther to the right. And then once these folks had unfortunately passed from the disease, which happens to everyone with Alzheimer's, we have no current trajectory modifying treatments for Alzheimer's disease. But once these folks had passed, they were able to take these sort of gold standard histopathological measures of how much tau had actually accumulated in those brain regions. And they showed that the amount of premortem alpha synchrony was strongly related to the amount of tau that would have accumulated in those folks' brains post-mortem. So just a really good example of a study that linked these changes in neurophysiology to the accumulation of these harmful proteins. And there's been several other studies that have done similar work in folks pre-mortem as well. In Parkinson's disease, in contrast, beta oscillations are much more sort of known to be important to the disease features. So the strength of beta oscillations are known to relate to motor impairments. They're known to predict the efficacy of a really common treatment called deep brain stimulation, which is an invasive measure that's known to be one of the best treatments for movement problems in Parkinson's disease. So there does seem to be different features of neurophysiology that are relating to each of these disorders. But again, we don't really have a strong understanding of what molecularly is actually leading to these changes in neurophysiology. So then if we want to dive into those questions and look at neurophysiology and some of the bases of these neurophysiological changes, the question then obviously comes up, why use MEG for this, right? There are other measures of measuring neurophysiology, so why focus on MEG? And I think there's really two ways to answer that question. The first one is kind of scientifically cool and what a lot of people focus on. And the second approach, I think, is something that gets overlooked a bit, but is equally important in my mind. So in terms of the technical advantages that MEG offers for studying these patient populations, obviously the thing that everyone talks about first is that MEG has a really unique combination of very high spatial and temporal resolution. This means that across the brain simultaneously, we can measure these really fast patterns of brain activity as well as slower patterns of brain activity non-invasively in these awake, behaving human participants, which is not really the case for other methods. In EEG, we have worse spatial resolution. In intracranial recordings, we have worse spatial coverage. And in fMRI, for example, we have much worse temporal resolution. So MEG really gives us the unique ability to measure with high temporal and spatial resolution neurophysiology across the human brain in a way that no other method does. Another thing that's really important here is that MEG gives us a direct measure of neural potentials. So the electrochemical potentials surrounding cells that are actually influencing their firing directly is what we're measuring with MEG, whereas in a method like fMRI, where we're focusing on the bold response, we're actually measuring hemodynamics. So in disorders like Alzheimer's and Parkinson's disease, where we know that there are these vascular changes that accompany the disorder and likely changes in the neurovascular coupling that leads to the signal of interest, there's a bit of a confound there where we never know if what we're looking at is truly changes in neural activity, or if we're instead kind of also incorporating these changes in vasculature into our measures. And then finally, and particularly in the case of Parkinson's disease, it's important to note that MEG is less averse than other methods to the presence of ferromagnetic implants. So this isn't to say that we're totally immune to them. Obviously, we're measuring magnetic fields here, so we do need to be aware of the fact that these magnetic implants are going to emit a magnetic field that's measurable with our sensors. But MEG is able to deal with these artifacts a bit better than other methods. And in some cases, in things like fMRI, these implants are actually exclusionary because they can be dangerous for patients to go into a scanner with these magnetic implants in their bodies. So in Parkinson's disease, this is particularly important because, as I mentioned before, one of the most common treatments for very advanced and very difficult to treat movement problems in these patients are deep brain stimulators. These are actually invasive electrodes that we implant in deep brain structures in these folks with Parkinson's disease, and it really helps a lot of motor symptoms in these folks. But again, it's this electrode that's magnetic, that's sending an electrical signal as well into these brain tissues in the patients with Parkinson's disease. And MEG is able to actually handle these artifacts much better than other methods. So a good example of that is on the right here. This is a paper from Ashwini Oswal et al. That was published in Brain a few years ago. And what they showed here that was really interesting is that they could measure coherence, so shared information, connectivity, so to speak, between these deep brain structures that were being stimulated by DBS and the cortex, the motor cortex specifically. And they were able to show that there were changes in the amount of communication between these two brain regions, specifically when the DBS system was turned on versus when it was turned off. So that's really a cool proof of concept, I think, to show that with MEG, we're actually able to look at brain activity even during active stimulation with some of these ferromagnetic implants, which really isn't the case with other methods. Now, another side of this that I think gets overlooked, but from my own experience in recruiting and recording data from patients with these neurodegenerative disorders, I think this is a really important thing to consider. MEG is also practically a much more comfortable experience for the participants than comparable methods of neuroimaging. So the MEG environment itself is quiet and less confined. You can see an image of a recent install from a MEG system over here on the right. You essentially sit in this relatively comfortable chair, you're raised up into this helmet, and you're in this much less restrictive sort of open room with the door shut. And compared to things like fMRI, where you need to sit inside of a relatively confined bore, this can make a real difference. So that, alongside the fact that there's less pre-scan preparation than things like EEG, when we talk about patients who are coming in who oftentimes have anxiety problems, some of them have severe memory deficits, so they may not remember where they are from the beginning to the end of the scan. This really contributes to them having a more pleasant experience in the scanner, but also to our ability to collect more interesting data and to collect more data. So in my experience, those patients with amnestic problems who are coming in and sitting in our scanner are able to do relatively difficult cognitive tasks with a high degree of accuracy that we may not have expected them to be able to do in other scanning environments. Great, so hopefully I've convinced you that MEG is a really great tool to study some of these neurodegenerative problems. I'm gonna dive into some of my own research in this area now. The first of these studies, as I mentioned before, is looking at how the accumulation of harmful proteins in the brains of healthy older adults, as well as patients with Alzheimer's disease, is leading to some of the changes in neurophysiology that we oftentimes see in these clinical groups. So starting with a little bit of background, this is another paper from Kamalini Ranasinghe and Srinagar Rajan down at UCSF. And they kind of started leading the way along with some other folks like Fernando Mestu, looking at how the accumulation of these harmful proteins might be impacting brain activity. You can see over on the left here, we have fast patterns of brain activity in the alpha band, and on the right, we have slower patterns of brain activity in the delta and the theta bands. On the top, we have accumulation of these tau proteins, and on the bottom, these amyloid beta plaques. So these, again, are two different proteins that are known to contribute to the progression of Alzheimer's disease and lead to cognitive problems. And what they showed here was that both these fast patterns of signaling and these slower patterns of signaling both overlap significantly with the amount of these harmful proteins that had accumulated. So they're seeing that the accumulation of these proteins might be leading to these changes in neurophysiology that we oftentimes see. Now, what you can't see from this plot, but is worth noting, is that these relationships actually go in opposite directions based on the frequency band that's involved. So these fast patterns of signaling are actually decreasing as more proteins accumulate, whereas these slow patterns of signaling are instead increasing as more of these proteins accumulate in these brain regions. So during my PhD work, I was interested in trying to sort of bridge that gap and model these two patterns of change simultaneously with a single model of what we're calling neurophysiological slowing, because again, it's an increase in slow activity and a decrease in fast activity. And what we were able to show was kind of a replication of this, that the more amyloid that we're seeing in the brain regions of these individuals across individuals with Alzheimer's disease, the more of these neurophysiological changes that we're seeing as well. So we're seeing a slowing or a hypoactivity of neural activity, which is more negative values on this x-axis, as we're increasing the amount of amyloid that's accumulating in these brain regions. Now, I'm going to focus on this plot on the left here because it uses an analysis approach that I'm going to be touching on throughout my the other studies that I'll be presenting today. For this analysis, when we wanted to look at how this slowing of neurophysiology was relating to the accumulation of these harmful proteins, we used what's called a linear mixed effects analysis. So essentially, each of these colored lines that you see in the background of this plot is one individual with Alzheimer's disease. And what we're doing is we're first at the sort of base level of this model, we're looking at the relationship across brain regions within that one individual between how much amyloid's accumulated in that region and how much neurophysiological change that we're seeing relative to the control group. So that's what each of these line means. It kind of tells you how much of a relationship there is between proteinopathy and neurophysiology in the brain of that one person. And then we nest those individual models within this larger linear model, which is represented by this overlaid black line, to see if there's a sort of a statistically significant and consistent trend in those relationships. So do we consistently see that there's a relationship between amyloid uptake and changes in neurophysiology? And again, that is what we see. We see that more amyloid accumulation is leading to more changes in these neurophysiological patterns across individuals and brain regions. And interestingly, what we were also able to show then, taking this one step further, by taking the relationship within each individual, by looking at this slope that you see plotted for each individual in these colored lines here, and using that as a measure of how much proteinopathy is impacting their brain activity, we could then actually show that those individuals who had more of a negative impact, more of a pathological looking impact of these harmful proteins on their neurophysiology, which would be more negative values on this x-axis, those were the same individuals who were doing worse on tests of attention function, on tests of cognition. So we're seeing that the combined relationship between proteinopathy and neurophysiology is itself also relating to how progressed these folks are going to be on tests of cognitive function in Alzheimer's disease. So this work and work from others has shown that there might be a relationship between the accumulation of these harmful proteins and these changes in neurophysiology in patients with Alzheimer's disease at the clinical stage. But the key question here, and something that I think is really important to consider, is that by the time these folks are showing up in clinic with memory problems, or even in the sort of early memory disorder problems of the disease, these folks have likely been accumulating these harmful proteins in their brains for 5 or 10 years, or maybe even longer. So what we know now from decades now of research is that it's unlikely that treatments or even the use of these neurophysiological features as potential biomarkers for prognostics or diagnostics, it's unlikely those things are going to be very useful at the clinical stage of the disorder. These folks have already been on what we're calling the Alzheimer's disease continuum for several years at this point, most likely. So the question that we need to answer then is, do we see similar relationships between the accumulation of these harmful proteins and these changes in brain function early on, when we actually might be more efficacious with treatments in the preclinical stages of the disease? So I was really excited when I got my postdoc here at the Montreal Neurological Institute working with Sivan Baye, because it allowed me to become a collaborator on this project called the Prevent AD Research Cohort. And what's really unique about this cohort, which is open science, most of the data has been openly released and it's still ongoing, so I really encourage everyone to check this out. But what's really unique about this data set is, in this data set, we're studying who these folks who, for all intents and purposes, are healthy controls. So these are folks who are asymptomatic. At the baseline visit, they're showing absolutely no signs of cognitive dysfunction or anything like that, that's signaling that they're on the Alzheimer's disease continuum. But they are at higher risk of developing idiopathic Alzheimer's disease based on their family history. And at baseline, they've been collecting task-free MEG data, so resting state MEG data. They've been collecting amyloid and tau PET data across the cortex, so being able to look at the accumulation of these proteins again across the brains of these folks who are cognitively healthy, as well as detailed neuropsychological and clinical testing. And importantly, that neuropsychological testing is also being repeated longitudinally for several years out from that baseline scan. So what we want to be able to do with these data is we can take those neuroimaging measures of neurophysiology of proteinopathy at baseline, see how those are actually relating to eventual prospective cognitive declines that might signal that some of these folks are progressing towards an Alzheimer's disease clinical phenotype. So before I dive into some of the actual research here, I do need to give a shout out to Jonathan Gallego-Rudolph. He's a PhD student who I'm co-mentoring here at the MNI with Savan Baye and Sylvie Villeneuve. All of the work that I'll be showing for the next few slides was him doing the heavy lifting. He did all the button clicking, and I was just there for the fun parts, building models and hypothesizing about things. But Jonathan and I and our team, what we needed to do before we started running analyses on these data was we first needed to understand what we were expecting in terms of relationships between proteinopathy and neurophysiological change in the preclinical stages of Alzheimer's disease. And this is really difficult because the vast majority, essentially all of the work that's been able to look at both amyloid and tau in the human brain in relation to these neurophysiological features has been conducted in the prodromal and dementia stages of the disease. So over here, including the studies that I just showed. And because of that, there's really no research telling us what we're expecting in the brains of these folks who are, again, for all intents and purposes, healthy at these baseline data collection time points. So we had to fall back then on animal literature and on in vitro cell line literature, where scientists have looked at how exposing de novo neurons to these cytotoxic proteins changes their function, changes their patterns of firing. And what we saw was kind of an interesting pattern and not necessarily what we expected, but something that I think is pretty intriguing. So we saw that in these animal models and in these cell line models, when you do expose these naive neurons to amyloid beta and to tau proteins, you don't actually initially see that shift towards slowing of neurophysiology that we saw in the clinical disease stages. What you instead see is this early pattern of hyperactivity, so hyperexcitability when you expose neurons to these proteins. And it's only when you get this combined sort of negative synergistic effect of both amyloid and tau proteins accumulating that you start to see death of neurons, you start to see loss of cell circuit integrity, and you start to see this shift towards hypoactivity and neurophysiological slowing. So what we were hoping in this study, when we wanted to relate, again, these proteinopathy maps to these changes in neurophysiology in these participants, we were hoping that we would be sensitive to this inflection point, that we would see this hyperactive pattern starting to shift towards hypoactivity in these folks who are preclinical or potentially not even on the Alzheimer's disease time course at all. And that's exactly what we saw. I'll step through these plots a bit more slowly because they look pretty complicated. But again, these are just similar models to what I was showing in that previous study where we were using linear mixed effects approaches to model the relationship between proteinopathy and neurophysiological change. So on the x-axis here, we have amyloid beta across brain regions and individuals. On the y-axis, we have neurophysiological change in these two different frequency bands. And again, each of these lines in the background represents one participant in this sample, and it's representing the relationship between amyloid and neurophysiology in that one person's brain. And what we saw across brain regions and across individuals was that brain regions that tend to accumulate more amyloid beta, which is going more rightward on this x-axis, those are the same brain regions that are showing less slow frequency activity and more fast frequency activity. So indicating this shift again towards a hyper excitability or acceleration of neural activity in the presence of more of these proteins. But more importantly, when we then split up that group, so when we split up these lines in the background based on those individuals who were accumulating meaningful levels of this other harmful protein, this phosphorylated tau protein, versus those who were not accumulating that protein, which is indicated by the red and the black lines here respectively, we saw a shift away from that hyperexcitable acceleration of neural activity with increased amyloid, and we saw a shift towards what we're seeing in the clinical disease stages in those previous studies that I talked about. We're seeing a shift towards a neurophysiological slowing effect in those individuals who have both amyloid and tau accumulating in their brains. So this indicates that those folks might be shifting towards this sort of Alzheimer's disease clinical phenotype, but of course, we can't say that without actually showing changes in cognition in the long term that are related to these baseline neuroimaging measures. So we saw that, in fact, the combined accumulation of tau and amyloid and its effect on neurophysiology does relate to eventual declines in cognition in a way that, again, indicates this pathological relationship. So this plot's pretty complicated, but essentially what we're looking at here on the x-axis is going to be the impact within an individual of amyloid on their neurophysiology. So more negative values are going to, again, look like a shift towards that more pathological phenotype. We're seeing that those folks with a more pathological looking relationship between proteinopathy and neurophysiology are the same individuals who are showing these longitudinal declines prospectively, so 5 or 10 years out, who are declining more towards an Alzheimer's-like cognitive phenotype. But importantly, we're only seeing that effect in those individuals who have both amyloid and tau accumulating in their brains, again, indicating that it's kind of this negative synergy between these two proteins on neurophysiology that's leading to these eventual declines in cognition. So to wrap everything up from that study, what we're showing here is that there seems to be these multi-frequency patterns of changes in cortical neurophysiology that do track with the sort of major hallmarks, proteinopathic hallmarks of Alzheimer's disease. And importantly, this tracks even in asymptomatic older adults, indicating this might actually be useful for as a prognostic tool or even a diagnostic tool in these early stages when that might be valuable. We also see that this tracks longitudinal declines or predicts longitudinal declines in the key cognitive features of Alzheimer's disease. So again, this indicates that we're actually tracking something that's going to differentiate potentially folks who may or may not eventually decline cognitively to a state that would be considered mild cognitive impairment or Alzheimer's disease. Okay, so switching gears a bit now, I'll go into a different set of studies that looks at a different kind of topic and a different disorder, but I think again uses a lot of the same ideas and shows how MEG can really be leveraged alongside these other neuroimaging methods to try to understand the bases of some of the changes in neurophysiology that we see in patients with Parkinson's disease. So for a bit of background here, we know now, again, from quite a bit of research, that there are several neurochemical, so neurotransmitter systems that tend to be affected in patients with Parkinson's disease. Probably the most well-known of these is the dopamine system. So everyone kind of knows that Parkinson's disease is related to dopamine dysfunction. The most common treatment for Parkinsonism is levodopa, which is a form of dopamine that's distributed across the brain and helps with motor problems in the disorder. And we know that these dopamine deficits are due to the death of those neuromelanin-rich cells in the substantia nigra pars compacta, which is this deep brain nucleus that you see shown down here. And this leads to aberrant or dysfunctional dopamine signaling along what's called the nigrostriatal tract. This leads to changes in beta frequency oscillations, like I mentioned before, which then leads to problems with motor function. So there's kind of this substantia nigra dopamine beta oscillatory pathway that's known to be relevant for the motor dysfunctions that are seen in patients with Parkinson's disease. Now, there's also been emerging evidence now that there's really strong degeneration relatively early in these patients as well in both norepinephrine systems as well as acetylcholinergic systems. So noradrenaline, norepinephrine, those are synonyms, so I'll use them interchangeably a bit here. But these changes in noradrenaline signaling are known to be due to degeneration of what's called the locus coeruleus. It's another deep brain sort of brainstem structure. So it's a very small cluster of cells. These are also neuromelanin-rich cells, which is interesting that we see degeneration of both of these sort of key neuromelanin-rich nuclei in Parkinson's disease. But rather than supplying dopamine projections, these locus coeruleus cells provide the vast majority of cortical norepinephrine in the human neocortex. So when we look at locus coeruleus degeneration, we're really looking pretty specifically at degeneration of the norepinephrine systems of the brain. And it's been suggested, but it hasn't been empirically shown, that degeneration of these norepinephrine systems might be related to some of the key cognitive problems that we oftentimes see in the patients with Parkinson's disease. So norepinephrine is really well linked to things like memory formation as well as alertness and attention. So it's pretty reasonable to think that these norepinephrine degradations might be leading to some of the cognitive problems we see in these patients. And then finally, I mentioned there's also been some research showing that acetylcholine function degrades in Parkinson's disease as well. This seems to be more variable and maybe more specifically related to what we call Parkinson's disease dementia. So it's more common in folks who are progressing to these really problematic and really severe cognitive dysfunction stages of Parkinson's disease. But this is due to death of cells in this basal forebrain nucleus called the nucleus basalis of Meinert. And again, this is one of those nuclei that supplies the vast majority of its respective neurotransmitter to the entire neocortex. So when we see degeneration of the nucleus basalis of Meinert, we can pretty confidently say that we're looking at actual degeneration of these cortical acetylcholine systems. So since we're seeing degeneration of these neurochemical systems that seem to be relatively specific in patients with Parkinson's disease, and we're also seeing these parallel changes in neurophysiological activity in the brains of these folks, something that I really wanted to look into was, do we actually see that these changes in brain activity are shaped by those neurotransmitter systems that we know are degrading? And even more importantly, do we actually see that those relationships are meaningful? Do we see that they're actually telling us more about these clinical declines in motor function and in cognitive function than if we just used each method in isolation? So to look at these questions, I've been working on another open science data set. Again, something to really look into. All of these data are openly available through what's called the Quebec Parkinson Network. It's been a really fantastic resource to work on. These are folks with idiopathic Parkinson's disease, which just means that we don't know where the Parkinson's disease is coming from. There are certain forms of Parkinsonism that do have very specific etiologies, but this is unknown origin Parkinson's disease patients. And what we have in these patients that's really unique is a really extensive battery of different neuroimaging methods, including task-free MEG, alongside things like MRI sequences like neuromelanin sensitive MRI, which I'll talk about a bit more here in a second, as well as detailed clinical and neuropsychological information, again, on this really large patient group that's been a really fantastic resource to work with. So looking at the brain activity of these patients with Parkinson's disease compared to a matched healthy control group, you can see the demographics of our groups up here on the right. We first just looked at whether we were seeing the sort of typical changes in neurophysiology, these deviations in neurophysiology that we would expect in patients with Parkinson's. And we do. We see that in posterior cortices, there's an increase in the slow theta frequency activity. There's also an increase in these slower bands of alpha in these frontomotor cortices bilaterally that you see here in the middle panel. There's also changes in what we call the aperiodic spectrum in these folks in sort of these ventral posterior regions. I won't go into what that means right now. It's something I'm more than happy to talk about in the Q&A section because I think it's a really interesting topic to dive into. But for the sake of time, I can't really go into rhythmic versus arrhythmic activity here. And in parallel with these neurophysiological changes, again, we're seeing degeneration of that locus ceruleus structure when we use something called neuromelanin sensitive MRI. So for our purposes today, we just need to know that this is an MRI sequence that measures sort of the structure of this very tiny neuromelanin containing nucleus. You can actually see these nuclei if you squint in this plot bilaterally here. And you can see that these little tiny dots are brighter in this healthy control example participant than what we're seeing in this patient with Parkinson's disease. And what we're seeing again is in these patients with Parkinson's disease compared to a matched group of healthy controls, we're seeing reduced integrity of that locus ceruleus structure. We're seeing less neuromelanin, which indicates more degeneration of those neuromelanin-rich cells in the folks who have Parkinson's compared to the controls. And when we relate these measures of locus ceruleus integrity to these changes that we're seeing in the brains of patients with Parkinson's in terms of their neurophysiology, we see a really specific and interesting potential sort of pathway between locus ceruleus degeneration and changes in alpha power. So those frontomotor changes, increases in alpha frequency activity that we were seeing on that last plot in these patients with Parkinson's disease seems to be significantly related to the amount of degeneration that they're showing in their locus ceruleus, indicating this might be related to norepinephrine dysfunction in the brains of these individuals. Interestingly, we also see that the strength of that relationship tells us whether or not these folks are experiencing stronger stronger impairments in attention function. So those individuals that have more of a pathological looking negative impact of locus ceruleus degeneration, increasing their alpha power, those are the individuals who were seeing the strongest problems with attention function and worst attentional test scores on. So I mentioned that this is indicating this link between locus ceruleus degeneration and alpha frequency increases might be kind of a new pathway that we're suggesting between locus ceruleus and norepinephrine, its impact on alpha activity, and then these impacts that we're seeing on attentional function. But of course, the missing link there is actual norepinephrine function. So what I wanted to do next is look at whether we're actually seeing these sort of pathological looking relationships between locus ceruleus degeneration and these patterns of alpha signaling in brain regions that are more sensitive to this specific neurotransmitter. Luckily, we have access here. Again, another open science data set that I'm gonna harp on a bit. The NeuroMaps data set is openly available. It's hosted here at the MNI. It's really fantastic. It provides these atlases of different neurotransmitter systems in the healthy human brain. So we can actually say, where in the brain do we see more of these, in this case, norepinephrine transporters for this specific neurotransmitter, which would tell us that those brain regions are more likely to be sensitive to that specific neurotransmitter, if that makes sense. And what we saw here was really interesting. We saw that those brain regions that are more sensitive to norepinephrine, that are showing higher density in the healthy brain of these norepinephrine transporters, those are the same brain regions where we're seeing the strongest negative relationship between locus ceruleus degeneration and alpha activity, indicating that this is likely a norepinephrine specific pathway. This is actually noradrenergic in nature. So from there, one thing that I wanted to keep looking at was, how do other neurochemical systems maybe shape these changes in neurophysiology that we're seeing? And this is a difficult question to answer. That's because we don't always have these really nice sort of neurochemically specific structures to look at. We don't always have good MRI measures that just happen to be sensitive to a specific pigment in those structures. So I wanted to take a more comprehensive approach next and look at whether we're seeing neurochemical systems shaping these changes in neurophysiology across several systems across the brain. So to do this, I first took the MEG data from that Quebec Parkinson Network data set. So a lot of the same individuals are included in this sample. And from the MEG data, we source imaged it. We did a spectral analysis across the entire brain and then resampled into an atlas. So these are pretty typical maps that you would see of neurophysiology for all of our participants, both our patients and our control group. We have these maps of neurophysiology in an atlas space for each of our frequency bands. So delta, theta, alpha, and beta, increasing in speed as we go down. And then kind of the neat thing that we did here is we took those maps, and for each patient, we standardized their brain maps within each frequency to the comparable data from the healthy control group. So what we get here are maps for each of our patients with Parkinson's disease that represent deviations across brain regions from healthy normal levels of activity at that specific frequency. So these are deviations, maps of neural deviations rather than just overall maps of neural power. And what we then did is we took those maps of Parkinson's disease related deviations in neurophysiology, and we looked at whether they spatially sort of preferentially aligned with these different maps of neurotransmitter densities that I talked about on that previous slide from this NeuroMaps database. So NeuroMaps includes PET data from several different groups, from several different samples across 19, I believe, different neurotransmitter systems, things like dopamine, serotonin, acetylcholine, norepinephrine, as well as some others. And to look at how these neurophysiological changes from Parkinson's disease are aligning with these specific neurochemical systems of the brain, we again use this linear mixed effects approach. So I have kind of a graphic that walks us through it here. Basically what we're doing here is, for each of these neurochemical atlases that we're interested in, each of these neurochemical systems, which is on the x-axis here, we're looking at the relationship to that neurochemical atlas across brain regions to the neurophysiological changes that we're seeing in each of our patients. So essentially, we're fitting a model within this larger model, showing us how much the changes in neurophysiology in each of these patients are actually lining up with different neurochemical atlases, different neurochemical systems in the brain. We do this for each patient. So we do it for patient 1, we do it for patient 2, and we do it for all of our patients. And then again, we nest these models within a larger nested linear model that tells us whether there's sort of a consistent co-localization of these brain activity alterations with specific neurochemical systems in these patients with Parkinson's disease. So I won't go through all the results from the study now because it's a pretty extensive study and there's a lot of different interesting findings here. But the one that I really want to highlight is this kind of interesting co-localization between broadband increases in brain activity and acetylcholine systems in the brains of these patients with Parkinson's. So what we're seeing over here on the left are the results of this linear mixed effects analysis. So you can see that in terms of the frequency band, we're seeing this increase in all of our frequency bands, beta, alpha, delta, and theta, that are relating positively to this specific neurochemical system that we're looking at. And the plot over on the right here shows the nature of that effect. And what we're seeing is that these deviations relative to controls, so remember these are always deviations relative to healthy levels, these broadband deviations, these broadband increases in neurophysiology tend to be specific to brain regions that are showing higher levels of these acetylcholine receptors and transporters, indicating that these might be specific, these changes in brain activity might be related to acetylcholine function in the brains of these patients with Parkinson's. And even more interestingly, we see that the strength of that co-localization, the strength of that alignment of these neurophysiological changes with these acetylcholine systems are actually also related to attention function, to how these folks are doing on cognitive tests. So the plot on the left here is similar to what we've seen before. These individual sort of lighter lines in the background are single patients' brains with Parkinson's disease. And again, the slope of those lines is going to indicate how strongly their increases in broadband activity relative to controls are related to these acetylcholine systems in the brain. You can see that we have these data also separated out here by attention function. So the blue line here indicates folks who are doing better on test of attention, and the red line indicates people who are doing worse on test of attention. And you can see that those individuals who have a stronger positive relationship are actually doing better cognitively. So this indicates that these broadband increases in neurophysiology that we're seeing in the brains of patients with Parkinson's disease, specifically in these acetylcholine systems, they might actually be compensatory. They might actually be helping people because those are the folks who are actually doing better on cognitive tests in this sample. So to try to wrap all of that together, basically what we're seeing is that these changes in neurophysiology and Parkinson's disease do seem to be systematically shaped by different neurochemical systems, by different neurotransmitter systems. We see that these broadband increases in power do tend to be stronger in regions rich in acetylcholine, whereas alpha increases specifically seem to be related instead to noradrenergic or norepinephrine dysfunction. And I think really interestingly, we see that the strength of these relationships tells us about the individual variability of these patients in terms of how they're presenting clinically. Specifically in terms of cognition, which was really interesting to me, we saw that these broadband acetylcholine relationships were related to preserved attention function, indicating that they may be compensatory in nature, whereas the relationship between alpha frequency increases and these norepinephrine systems indicates the opposite in the same test of attention function. We actually see that more of this pathological relationship between locus coeruleus degeneration and norepinephrine systems, and alpha frequency changes is actually leading to impairments in attention and worse attention function across individuals. And then finally, taking a step back, I think what these studies tell us in whole is that the topography of these functional changes in the brains of patients with Parkinson's disease are also meaningful. So oftentimes when we're doing these studies of these patient groups versus control groups, the first step we take is we do a comparison between groups, and then we only focus in on brain regions where we see statistically significant, based on frequentist inferential stats, these statistically significant effects that are different between groups. But this tells us that we might be throwing out a lot of meaningful information when we do that initial thresholding step, kind of a priori without a lot of basis for it. So finally, to try to wrap all of this together with a nice bow for the holidays, I think what this tells us as a whole, what all of these studies tell us is that we can combine MEG with direct measures of things like neurochemistry, proteinopathy, and structural integrity of different brain regions to really understand the molecular mechanisms that are contributing to the brain activity changes in these states of disease that we're oftentimes so interested in. I think it also tells us that we can use these multimodal fusion types of studies to identify what targets there might be for therapeutic intervention based on what's actually impacting neurophysiology and what's relating to these key clinical features. So things like pharmacological interventions and neuromodulatory interventions might be guided really effectively by combining MEG with these other methods to try to really understand what's happening at the molecular and the structural levels of the brain to lead to these changes in neurophysiology that might be a bit closer to the actual clinical features of the disorder. And then finally, taking one last step back, I think the really key thing here is that this work and other studies like it show that we can potentially use MEG as a really strong, repeatable, non-invasive prognostic and diagnostic marker. So because it's so non-invasive and because it's so such an easy scanning environment to put people in, MEG could serve as this really nice, sensitive and repeatable way to try to understand these diseases in the preclinical stages and to use as this kind of marker. And it also might be more sensitive than many of these other features that I've been talking about today. So things like deep brain structure, these scans of deep brain structure, while they're fantastic and they give us this really interesting information, we might expect neurophysiology to change on a faster time scale along these disease processes than something like structure. So cells dying might happen in a way that's detectable on these scans at a time scale that's much slower than these neurophysiological changes. So MEG might represent a really fantastic tool clinically as well, based on this research. So with that, I just need to thank everyone involved in this, specifically Sivan Baye, whose lab I'm working in right now, as well as Jonathan Gallego-Rudolph and all of our collaborators on the Prevent AD cohort, our funders, as well as our open science research initiatives that really made all of this possible and saved me from a COVID data analysis slump. So I have huge thanks for them. And with that, I'll take any questions.
Hi there. Yes, so we are here for the Q&A portion. We have a question. Does the U-shape relationship observed for the physiological features, alpha power for instance, in the preclinical stages of AD, limit the efficiency of these features in early detection / prediction of AD in individual subjects? Thank you so much for the great talk.
Yeah, that's a fantastic question. I think it's something that we've been thinking about on our end as well, because if we're looking at these neurophysiological measures and we're seeing, for instance, an increase in this alpha activity, is that pathological because it's relating to the accumulation of this amyloid protein, or is that actually indicating someone who's dealing with those proteins better, at least for the time being? And I think that's something that we've been looking into, and we're seeing that it is actually potentially that second option. So what we're seeing is, if we just take in a new study that we've been running that I didn't talk about here, we've been looking at how we can use these MEG features alongside sort of basic, easy to collect clinical features to predict who's going to decline cognitively in the long term. So not looking at shared relationships here between MEG and neurophysiology with these proteinopathic markers, but just looking at it individually. And what we're seeing is those individuals who have this acceleration of like relatively accelerated brain activity compared to their peers, those are the folks who are less likely to cognitively decline in the long term. We think that's because amyloid proteins, and I didn't go into this maybe as much as I should have, but amyloid proteins accumulate in a lot of folks' brains who will never actually progress to an Alzheimer's disease phenotype. So oftentimes you will, if you put people who are cognitively healthy in the PET scanner and you get this amyloid map in their brain, you'll see elevated amyloid levels, but that person will never actually develop memory impairments and never, sort of progress to a dementia stage or even a mild cognitive impairment stage. So there's also this really interesting line of research showing that in healthy adults, when we're not considering proteinopathy or anything, we actually see an acceleration of neural activity there as well, just with healthy aging. So what we're thinking is that essentially this acceleration of neural activity might be this sort of beneficial compensatory mechanism showing that folks are going to hopefully be dealing with these harmful proteins better. But then once that other protein starts accumulating, that tau protein starts accumulating, those are the folks who are going to be shifting towards sort of a neural slowing phenotype, and those are the folks who are going to potentially be much more likely to progress towards MCI and Alzheimer's disease. So essentially, if we're seeing this relative acceleration of neural activity, we see that as either beneficial or not necessarily a negative, even if it's in the presence of amyloid. But it's really once we see that shift towards slowing that we're saying that looks more pathological. And there's work from other groups that's coming out as well, showing very similar things. But yeah, great question.
Yeah, thanks for answering that one. So someone else has written in a comment, thank you for the nice presentation. We agree. We really enjoyed this. And then another one is, do the broadband effects reflect changes in the same direction in oscillatory activity in several distinct bands, or do they reflect changes in the aperiodic component?
Yeah, so I'm glad someone asked about aperiodic features, because it's something that we've been really diving into a lot in our work. And I struggled with whether or not to include it in this talk because I think it's such an interesting topic, but it also is a bit more niche to folks who are really deep into the analytics here and the sort of mechanistic interpretations we can draw from this. It looks like it is an aperiodic effect. So I didn't dive into this as much, but that second study that I showed was actually done before the first one, and for that second study, we did not parameterize the spectra. So what that means is when we're looking at these neurophysiological spectra, for folks who aren't as familiar with this, we oftentimes see this sort of what's called a 1/f fall off, where as you get to faster speeds of brain activity, you're seeing less power in the signal. And oftentimes that sort of decline in power as a function of frequency has been treated as noise and background signal that no one is interested in previously, but more recently people have been showing that it's really interesting. It actually has a lot of interesting relevance to aging, to disease features, to cognitive function, even in healthy young adults. So there seems to be something embedded in that 1/f aperiodic pattern of activity that's in the background. So what we can do is we can do something called parameterization of the spectra. So we can take a model of that 1/f fall off, which follows a log linear relationship. We can model that within the brain data that we have, and we essentially separate out that sort of log linear straight line model, is what you can think of it as, from the bumps on the spectra that we think represent this more rhythmic periodic activity, just based on how Fourier transforms are computed. So in the study that I showed that related acetylcholine systems to these broadband increases in neurophysiology, we did not inherently correct for the aperiodic before running that analysis. So the fact that it's broadband like that indicates that it's almost certainly aperiodic. It's very unlikely that we would see meaningful rhythmic activity in all 4 of those frequency bands that were all going in the same direction in relationship to these acetylcholine systems. And actually, interestingly, in a follow-up study that I'm working on right now, we've also been using diffusion weighted imaging of that nucleus basalis of minor structure that I talked about. So it's a way of getting at the integrity of that basal forebrain structure that again supplies the vast majority of acetylcholine to the cortex in the human brain. And we're seeing a really similar shift specifically in the aperiodic. So we are parameterizing for that analysis, and we're seeing that degeneration of those acetylcholine systems from the basal forebrain are in fact related to a shifting of the aperiodic. So it does seem to be an aperiodic trend that's related to acetylcholine, whereas interestingly, the locus ceruleus alpha frequency relationships that we were seeing are in fact rhythmic. So that was also a parameterized study where we saw that those were specifically related to the rhythmic features of alpha frequency activity.
Thank you. So we still have time for people to write in questions, so feel free to do so. Here's another one. Have you considered how functional connectivity might be affected by these proteins and chemical changes in patients with neurodegenerative disorders?
Yeah, that's a fantastic question. So there's a few answers to that. First of all, there's been a lot of work done by other groups that have looked at connectivity in the context of especially Alzheimer's disease, but also in Parkinson's disease. So for Alzheimer's disease, I'd recommend looking into the work, especially of Fernando Mestu, of Kamalini Ranasinghe, and Sri Nagarajan at UCSF, as well as some others. There's Cornelis Stam as well for some of that work. There's also been a lot of work in Parkinson's disease looking at connectivity. So there's been great work from Elizabeth Heinrichs-Graham, as well as Cornelis Stam and a few others looking at Parkinson's disease features as well. We have looked a bit at connectivity in these studies, and we're starting to dive into that more. Our approach has really been to look at the basic sort of local neurophysiological features first, and then see at a second stage whether connectivity adds new information to predicting clinical phenotypes and to relating to things like proteinopathy or neurochemical systems. I don't want to spoil anything, but the PhD student that I'm co-mentoring is doing some really interesting analyses showing that the functional connectedness of different brain regions predicts how much tau is going to accumulate in those brain regions, and it's specific to certain frequency bands. So that's using some of those longitudinal data from the PREVENT-AD cohort that is still being processed and still being analyzed. So I didn't speak to that too much today, but it does seem like connectivity plays a really key role here in the Alzheimer's literature. And I think that fits with a lot of research that's been done using other modalities and in animal models as well. There's this idea that connected neurons are more likely to share these harmful proteins with each other. There's what's called a prion-like spread of these tau proteins within neurons. So there's a lot of research showing that might be the case here. So we think that connectivity might be playing a really key role here that's mechanistically interpretable as well. And then as far as in Parkinson's disease, I haven't looked at that as much, but it is something I'm really interested in because I think that kind of the key next step is, we're seeing dysfunction in these local circuits. Do we see that local dysfunction is also propagating to other regions, and how that region signals communicates with the rest of the brain? So yeah, lots forthcoming with that, but nothing to show in terms of data right now.
Thank you. Do you think that these relationships might be different if you considered MEG data recorded during performance of a cognitive or motor task?
Absolutely, and that's something that, again, I didn't dive into it today, but there is task-based data that I collected during my PhD research in clinically defined Alzheimer's patients, and we've shown that some of these patterns don't necessarily track perfectly into folks who are actively performing cognitive tasks, or in some cases they even reverse, where we see, for example, greater acceleration of neural activity during active task processing uh, uh, in the brains of these folks with Alzheimer's disease. So that's another future direction that I think is really important here. We're looking at task-free MEG in a lot of these cases. That can be useful when we're talking about things like biomarkers for diagnostics, prognostics, or even trying to normalize these patterns with things like non-invasive neurostimulation. But I think really the key missing link here is there something in the middle that's different when people are actually actively processing information, when they're trying to perform a memory task in Alzheimer's, or when they're trying to perform a movement in Parkinson's or things like that. There is some literature on that already, but it's something that I think needs a lot more attention, because these resting state studies are more feasible to collect data in from these patient populations. But at the same time, we're kind of missing a key piece of what's actually happening when these people are trying to think in the ways that they're impaired, because task-free imaging, it's maybe not technically a resting state, but it's certainly not during the active processing that we're oftentimes interested in relating these brain features to.
And I think this is going to be the final question. How do you see these findings translating into clinical practice?
Yeah, so I think with regards to clinic, the clearest and most sort of proximal thing that we could do with these data is we could start looking at, and again, we're starting to do this on our end already, start looking at how useful these different neurophysiological measures might be for prognostics, for diagnostics. I mentioned that study before where we're actually looking at how changes in neurophysiology at baseline are predicting who's going to convert to mild cognitive impairment and eventual dementia relative to those who will not. And we're seeing some really promising things. We're seeing that MEG does provide sort of independent and very useful information for those kinds of prognostics. So I think that's really the first step, is using this as a tool to understand who's going to progress versus who's not. The other approach that I think is interesting but a bit farther away, but still very promising, is this idea that we might actually be able to normalize these aberrant patterns of brain activity using things like non-invasive neuromodulation. So there's things like sensory entrainment or non-invasive neurostimulation with TMS or with transcranial electrical stimulation that we might be able to use to try to normalize these patterns of brain activity and see if we can help folks sort of regain some of their function in the short term. So an example of that would be in Alzheimer's patients. Maybe we can actually accelerate, so to speak, their neural activity in those populations that are slowing, with the hopes of rescuing some of those cognitive declines that we're seeing relate to those features. But again, that's something that I think is going to take several steps of research to make it to the clinic, just because there's a lot of groundwork to do in understanding what exactly to target, which we've been working on, but also how to target it, what the best method would be, and whether it's actually going to give any clinical benefit, because there's obviously the question of, if we're seeing slowing of neural activity and it's related to loss of cell circuit integrity, how much can we rescue function in circuits that have already lost integrity? So it's a really interesting topic, and it's something that we're actively pursuing and that I'm actively pursuing in my research, but it's something that I think is a bit farther away from using MEG more as a diagnostic and prognostic marker itself.
Thank you very much for that answer. You've also had a few comments through the chat to say thank you for the excellent presentation. So we at Megan would also like to reiterate thank you so much for being a presenter of this masterclass. It's been an excellent session. And thank you everyone online who's joined the masterclass today. And we hope to see you on future masterclasses next year. So thanks very much.
Great. Yeah, thank you so much for having me. It's been great.
Thank you.